I thought what we could do before we start talking about photorespiration and the last little bit of photosynthesis is we could go over both the light and the dark reactions just contiguously so that you guys can see demonstrated photosynthesis from the beginning to the very end, actually making sugars. So let's begin here, and this is the part that we've already gone over a number of times, so I'm sure you're comfortable with it at this point. But one more detail that I would like to add to the story before we move on is the relative sides of the membrane. So what we're going to be thinking about photosynthesis today, particularly with respect to the light reactions, is where within the chloroplast these things are taking place. Now remember, a chloroplast looks something like this. Right, it has an outer membrane, looks like a big Hershey kiss kind of, and then an inner membrane. And then inside that membrane, there was another membrane, a third series of membranes, and that was called the what? Thylakoid. Yeah, it's the thylakoid membrane. And the thylakoid membrane is actually the membrane that has chloroplasts embedded into it. And so when we talk about the light reactions, really we're talking about interactions and chemical reactions that are occurring on that thylakoid membrane. Now, when we're pumping protons here at the electron transport chains, what we're doing is we're pumping protons from one side of this membrane to the other. And the net movement, the active movement of protons is going to be from outside the thylakoid to inside the thylakoid. And so on this diagram here, the stuff up here, this is inside the thylakoid, and this is outside the thylakoid. Okay, so please keep that in mind. And if you're asked to diagram this on the exam, please make sure you indicate which side of the membrane things are occurring on. Okay? You okay with that so far?
Wait, so where's the side?
On the diagram, those green squigglies that are extending out.
Mm-hmm.
So that's the membrane. This is the thylakoid membrane. And the stuff up here, this is inside the thylakoid. And the stuff down here, this is outside the thylakoid. So it's like photosynthesis is occurring right here. Okay? No, it's just fine.
I got confused. So where the proton comes in is outside the thylakoid, and where like the electrons exit are inside?
So when we're pumping protons from this side into here, we're pumping protons into the thylakoid.
Okay.
Okay, so let's do this. Let's diagram photosynthesis and think about it in the context of where in the chloroplast this stuff is happening. Okay, so the first thing to happen is what? A photon, exactly right. So a photon comes along and it strikes a chlorophyll molecule. When it strikes the chlorophyll molecule, what happens to that chlorophyll?
It's excited.
It's excited, which means what?
The magnesium plates.
Yep. An electron on the magnesium ion
Pushes it out of the
jumps out an energy shell, right? So it moves a little bit farther away from the nucleus. And that represents some potential energy, right? The distance between the nucleus and the electrons. So we increase the potential energy of that magnesium ion. Then it falls back to ground state. It loses that potential energy, and that potential energy gets passed between chlorophyll molecules. And each one successively gets excited, right? Until eventually, that's passed over to this chlorophyll complex that I've drawn unfortunately small right here, and that's called what? P680. And P680 gets so excited that what happens to it? It loses an electron, right? So an electron is lost from P680. Yep, Kira.
So that's oxidation?
Correct.
Okay.
That's an oxidation reaction because an electron is lost. So P680 has been oxidized, and it's going to be reduced. Yeah, Adam.
Is P680 also being reduced by water?
It will be reduced by water. Exactly right. So water molecule comes along, and which side of the water molecule, or which side of the membrane is the water molecule on? Outside. So outside the thylakoid membrane, that happens, right? So a water molecule gets torn apart, and we produce some hydrogen ions, or protons, and then some oxygen gas which eventually just leaves the plant, hopefully. Okay, so P680 was initially oxidized, it lost an electron, and then it was reduced by water, right? So P680 gets reduced by water. And that electron goes up to the primary electron acceptor.
Question,
Yep.
on the quiz, there was a question about that, that said what reduces P six
It's
eighty?
number 6, it says which of the following is reduced by P680? And if you put water, it was.
It got marked wrong.
Yeah.
Let me check into
that
that.
happened to me too. Yeah, and I was like so confused.
I will check into that. I probably just mismarked the key. Thank you
There
for
were
bringing
two
that to my
answers, because you could say that, P700 is
So
also
run with
reduced
the-
by it.
Well, there were,
Okay, I'll look into that. And then if it sounds like I did, I made a mistake, then I'll make sure you guys get the points for it. Okay, so that electron reduces the primary electron acceptor, right? And it doesn't stay with the primary electron acceptor for very long. Eventually it gets passed over to our electron transport chain. So the electron transport chain, that first cytochrome protein, gets reduced by the primary electron acceptor, right? And then that electron, in a series of redox reactions, is passed down the electron transport chain. And for right now, we're just going to show it dropping out. And then what we're going to do is we're going to take protons, and we want to pump those protons to inside the thylakoid space. So you guys, what is the purpose of an electron transport chain? To produce a proton gradient, that's all it does. Just produces a proton gradient so that we can, using chemiosmosis, produce ATP, right? So we're going to take our protons and we're going to pump them from inside the from outside the thylakoid to inside the thylakoid. Okay, so we have a strong proton gradient inside the thylakoid space, right? So strong proton gradient inside here. And naturally, those protons are going to want to follow their concentration gradient and go back across the membrane. And the protein that allows them to do that is this, ATP synthase, right? So our protons are able to fall back through ATP synthase and fall out. So when protons fall through ATP synthase, what happens to that protein? It spins, which is a form of kinetic motion, right? So we're able to take that kinetic motion and we're able to take an ADP, which is adenosine diphosphate, or that ATP molecule with just two phosphates on it, plus another phosphate, and we're able to stick those two things together to make ATP. That's a really important product of the light reactions. Okay, so we have one of the products right here, ATP. So on the exam, when you're asked, the electron transport chain following photosystem 2 is responsible for creating which of the following molecules, and I give you ATP, I give you pyruvate, and I give you NADPH and other things, you're going to say ATP. Yeah. This is responsible for creating the proton gradient that makes ATP. So same thing happens with, and which photosystem is this? Photosystem 2. So it's photosystem 2, which means that this one is photosystem 1. Okay, so photosystem 1, same thing happens, right? We get we get electron or chlorophyll excitation by a photon striking a chlorophyll molecule. And that energy is passed between chlorophyll molecules, eventually over to P700. And then P700 loses an electron, becoming oxidized or reduced.
Oxidized.
Yep. Oxidized. And what is it that reduces P700?
See, that was the question, it said, what is reduced by P680, so would that be P700?
Yeah, if it's reduced by P680,
I
then
think
it's
that's what the
P700.
question said
Oh.
on the quiz.
So P700 is reduced by P680. Does that make sense? I see some consternation. So let me draw my arrow and then we'll talk about that for just a minute. So both P680 and P700 get reduced, right? And they're reduced by different molecules, and they're reduced after they become oxidized. Oxidation occurs when P680 and P700 pick up that energy from the chlorophyll molecules. They get so excited that they lose an electron and they're oxidized. And so the molecule that reduces P680 is water. It's the source of the electrons, right? And so the source of the electrons that's reducing P700 ultimately is P680. Does that make sense? You okay with that? Now, in reality, what I've shown is that these electrons are like all over the place. They're in and out of membranes and stuff. But in reality, this membrane kind of encompasses all of these proteins, and they stay mostly within the membrane. Not that that's a terribly important caveat for this discussion. But anyways, P700 gets reduced by the electron from P680 that's fallen through the electron transport chain. Okay with that idea? Okay. So our primary electron acceptor is reduced by P700, and then it passes that electron over to the electron transport chain, which has a series of redox reactions. And that electron just falls out the bottom. And again, we're going to use that electron transport chain to pump protons, right? Ultimately, that's all they do. Those cytochrome proteins and viridoxine proteins take protons from one side of the membrane, and they pump them through to the other side of the membrane. And which side of the thylakoid membrane are these protons being pumped to, inside or outside the thylakoid? They're being pumped from the outside to the inside, right? They're moving inside the thylakoid membrane again. Now, at this point, what I told you was there are really two fates for these protons. Either they can be cycled back and help create more ATP synthase and turn that, or excuse me, they can create more ATP by turning that ATP synthase a little bit more, or they can be used in this enzyme called NADP plus reductase. And so in this diagram, that's what we're going to show. We're going to show those those protons being used in the reduction reaction of NADP plus. Okie dokie. So these protons are going to kind of get picked up by this enzyme. And what this enzyme does is it takes NAD plus, and then it adds to it a proton and an electron. And we're going to make from that NADPH. What's the purpose of NADPH?
reaction.
Yeah, it just moves electrons. Adam?
I was gonna say it supplies electrons for the reduction of CO2 in the sugar.
Yeah, exactly right. So this is the source of the electrons of that reduction reaction where we take carbon dioxide and we actually make some sugars from it. And that occurs in the dark reactions that I'm going to diagram over here. Okay? So you have a whole bunch of ATP and a whole bunch of NADPH that's created. And remember, the full reaction for photosynthesis, at least the full reaction that I want y'all to know, is carbon dioxide plus, and there's 6 of those, plus 6 waters, gets converted into some oxygen gas, and ultimately a sugar, right? And so that whole equation diagrams really 2 separate reactions. The first of which is this reaction where we're taking water and we're converting it into oxygen gas. Is that an oxidation or a reduction reaction? Oxidation, which means it's what? Exergonic or endergonic?
Endergonic,
Close.
exergonic.
Yep. It's exergonic. It's releasing energy, right? Because we're taking something relatively complicated, a water molecule, and we're making something a little bit simpler from it. So that's inherently an exergonic reaction. The other reaction is this one. And it's a reduction reaction, right? Where we take carbon dioxide and we reduce it into sugar. Is that endergonic or exergonic? Endergonic, because we're taking something relatively simple and we're investing a whole bunch of energy into it to make something relatively complex. Now, where do we get the energy to drive that reduction? Close. It's the other one. ATP. That's the energy, right? ATP is the energy, and Adam, the source of the electrons, is NADPH. Exactly right. Okay, so this stuff that we're making here, NADPH and ATP, they're driving this reduction reaction. It's the energy and the source of the electrons. And we get those through this oxidation reaction, eventually. Are we okay with that idea so far? Okay, so we're going to take all this stuff